So here we have a human primary epithelial cell, proximal tubule cell in fact, and it's been treated for 2 hours with a mitochondrial decoupling agent called CCCP. Now this is a proton ionophore which will dissipate the inner membrane proton gradient in the mitochondrion. When that happens, the mitochondrion can't really make ATP anymore, and it starts to make a lot of reactive oxygen species, and it's quite dangerous for the cell, and the cell has to find some way of eliminating them. And so what I'm suggesting is that this might be a new way of eliminating mitochondria apically. So what we have here is what I think is a structure called an FR, or a fairy ring. Now it's called this because you see there's a ring of mitochondria here in blue, and so this is to heat shock protein 60, mitochondrial marker. Then you've kind of got a core of polycystin 1 in green here. This is 7E12 IgG1, and then there's an increased amount of Cu62 right in the very center of it. And so as you can see, this has only been going for about 2 hours, and it's about 4 microns across by about 4 microns. So it's quite a large structure. So it's a bit too big for, well, it's a lot too big for autophagosome, and it's much too large for a multivesicular vesicle. And so we think it's probably a new structure, and we call them FRs, or fairy rings, because when they develop, you have a green ring around around a core of Cu62, and embedded in it are kind of mitochondria that look a little bit like mushrooms. And so when we first saw them, we thought they were an artifact, but then we showed that they weren't, and that they were real structures. We verified our antibodies, and they only come on when you actually poison the mitochondria with a decoupling agent. So we'll quickly run this video and I'll comment on it. So here we're zooming in on it. So just a reminder, the blue are the mitochondria, green is polycystin 1, and red is Cu62. So we're just going to look at it from the bottom now, the base of the cell. So you can see here there's a kind of ring of mitochondria. So we just remove the mitochondria, and you can see there's a kind of core of Cu62 and polycystin 1 interacting with one another, we think, in close proximity. Now they won't co-localize because of the size of the molecules. Polycystin 1 is probably 50 to 100 nanometers long. Cu62 is binding to its PKD repeats, and the antibody 7E12 is to the very end terminus of polycystin 1, so it's some distance away, and so you wouldn't expect co-localization, but you would expect proximity, just like this. And we've taken the polycystin away, and we've got the mitochondria back, and you can see the Cu62 in red. And then we've taken Cu62 away, and you can see the polycystin 1 and the mitochondria associated with it. So this would be polycystin in the endoplasmic reticulum interacting with defective mitochondria, because these have been damaged with CCCP. Not too much, but just enough to decouple them a bit. 2 micromolar. Okay, and then you have this ring of mitochondria that look rather swollen. And around, we've got fragments of mitochondria that you don't see in normal cells. And then add it back into C62. Red.